This is a short tutorial on how to do the sex-linked inheritance lab for our biology class. So here's the background. We inherit many traits from our parents through the genes that they pass on to us. Some of those genes are located on the sex chromosomes, the X and the Y. These genes are called sex-linked genes. A female inherits an X from her mother and an X from her father. A male inherits an X from his mother and a Y from his father. Most of the sex-linked genes are located on the X chromosome. Two of these genes are for hemophilia. This is a disorder in which a person doesn't have the correct proteins to clot their blood. The disorder is inherited on the X chromosome. If a person only has recessive alleles for blood clotting, they will present the trait. The genotypes are XhH, or XhY. If a person has a dominant allele, they will have normal clotting blood. Color blindness is also a genetic disorder carried on the X chromosome. If a person only has recessive alleles for color blindness, the person cannot distinguish between certain colors such as red and green. If you have at least one normal dominant gene, you see all colors. So that's what this lab is about. We're going to toss some pennies and see how these are inherited differently in males and females. So I'd like you to go ahead and complete this Punnett square. We'll do this one somewhat together. If you look, this is a family that carries hemophilia. The parents, the mother is a carrier, so she has one dominant allele and one recessive, and the father has a dominant allele, so he is normal as well. So if you look, the mother, her allele split up. Notice that the H stays with the X, and then the father. As you combine these, the capital dominant alleles will always go first, such as in this carrier female. And then we have the two boys. So when you look at this, we just need to figure out, do they have the trait? And remember, this is recessive, so an individual must only have recessive alleles to show the hemophilia. So number of males that have normal is 1, and number of males with hemophilia is 1. Both girls are normal because each of them has at least one dominant allele. So make sure you copy this down, and you can go on and complete the other 3 Punnett squares that are shown in this packet. So here are the other 3. You have family 2, which has hemophilia. The change is that the father is a hemophiliac. Family 3 has color blindness. The parents are, the mother is normal and the father is colorblind. Family 4, the mother is a carrier and the father is normal. So you might want to pause this video so that you can get these completed before we actually create the pennies. Now for the pennies, we usually keep them over the turtle in the cabinet, so you'll need pennies and masking tape. So the general procedure is for each family, you will be shaking up 2 specific coins in your hands and dropping them on the lab table. The markings on the coins will tell you the genotype of the child that was just created. You're going to record the genotype with a tally in the appropriate box. You'll create 40 children for each family, so that's tossing the pennies 40 times. So here would be how you make the pennies. 2 pennies per group of 2 people, and about an 8-inch strip of masking tape to cover those pennies up. No groups of 3, you can work alone. Wrap the pennies in the masking tape so you can label each side according to the images above. So you can see that family 1, here's the first penny. On one side you're going to have the capital H, and on the other side you'll have the X little h. And the second penny, this is for the father, X with a big H, and the Y. Okay, so each family here is represented. What you'll want to do is maybe on the chalkboard write who is doing what family. Start with a different family. There might be repeats, but you'll eventually be sharing the pennies. You'll swap out so that you can do the other families. I'll come back to this in a minute so you can see it. Just looking at, make sure that you're going to enter the family data into the correct data table. So you make the tally marks and you total it up. Okay, so I'll go back and leave it here. You can pause so that all students get their pennies made and continue with the lab. And there are questions. Make sure you refer back to the introduction.